previous classes we have discussed about mitosis, a type of cell division. The next type of cell division is meiosis, which occurs in the germ cell to form the gametes, and the chromosome number gets reduced in this phenomena. That's why it is also known as reductional division, and it is occurring only in the germ cell and for the gamete formation, so that the chromosome number should be maintained from one generation to other. Now we'll discuss about the different events of this meiosis. It includes two sequential karyokinesis and cytokinesis, but the DNA replication will take place once only. Now meiosis 1, it includes two steps, that is meiosis 1 and meiosis 2. Meiosis 1 having 4 steps like the mitosis only, that is prophase 1, metaphase 1, anaphase 1, telophase 1. Whereas meiosis 2 includes prophase 2, metaphase 2, anaphase 2, telophase 2. But here in meiosis 1, the prophase 1 is the most longest period duration phase, and it involves 5 steps, that is leptotene, zygotene, pachytene, diplotene and diakinesis. That will study one by one what are the effects occurring during these stages. Depending upon the behavior of the chromosome, there are 5 steps, 5 phases in prophase 1 that we'll discuss here. Prophase 1 is the longest phase, and depending upon the chromosome behavior, how the chromosome is behaving in different stages, these stages are given their name. First is leptotene. When the leptotene starts, then the chromosome will undergo the process of condensation. That means the length of the chromatin material will become less, it will shorten as well as thicker, so that it will attain the identity of chromosome. It becomes condensed, compact and attain the identity of chromosome, and it becomes visible like a chromosome. Now it will enter into the next step, that is known as zygotene stage. In zygotene, the chromosome starts pairing. Two different chromosome, that is not homologous chromosome, will undergo the process of that pairing, and that will give rise to the synaptonymal complex, and this phenomena is known as synapsis. So synapsis is the identifying feature of this stage, that is zygotene. The pairing occurs between the homologous chromosome, and a pair of synapsed chromosome is known as bivalent, and when it will bifurcate, then it will be known as tetrad. So tetrad formation will take place during this zygotene, that we'll discuss through the diagram also. Then pachytene. In pachytene, the tetrads are fully appear, and the recombination nodules will appear. When crossing over will take place at these places, that is known as a recombination nodule, and the crossing over process phenomena will occur in pachytene stage among the two non-sister chromatids, where they exchange their genetic material, and that is helped by the enzyme known as recombinase, that will also discuss through the diagram. Then diplotene stage. After this pachytene, it will enter into the diplotene stage, where the synaptonymal complex which was formed during the zygotene stage will get dissolved, and the bivalent separate which was formed here, the bivalent separate except at the site where the crossing over took place, and that will appear like X-shaped structure, and this is known as chiasmata. This duration of this diplotene may be longer also in case of some vertebrates. In oocyte, it will be maybe 6 months or years also. Then it will enter into the next step, that is known as diakinesis. Here the chiasmata will move towards the terminal position of the chromosome, and it is known as terminalization. So this diakinesis is identified by the termination of the chiasmata, and finally they will get separated, and now the chromosome will undergo the fully condensation. If it is not, then nucleus, nuclear membrane, Golgi apparatus, endoplasmic reticulum will disappear, and the spindle fiber formation will start. Now the all the stages will complete of this prophase 1, Then it will enter into the next step, that is known as metaphase 1. In metaphase 1, like mitosis only, here the centromere of the chromosome will be attached with the spindle fiber, and all the chromosomes will be get aligned at the equatorial plate or the equator of the cell, and in this way their kinetochore or the centromere will be get attached with this spindle fiber, and finally they will form the equatorial plate. Then it will enter into the next step, that is known as anaphase 1. Here again, in the previous one, like the centromere will get splitted, the length of the spindle fiber get reduced, and half of the chromosome with half of the chromatids will be pulled towards the two opposite pole, and finally they will reach to the two opposite pole, that is south pole and north pole. Then it will enter into the next step, that is telophase 1. In telophase 1, like that only, the nuclear membrane will reappear, nucleolus will reappear, then the chromosome will undergo the process of decondensation, and it will lose its identity, and it will convert into the chromatine material. Golgi apparatus, endoplasmic reticulum, all will appear, and the cell having two nucleus that will undergo the process of cytokinesis to give rise to two daughter cell. Now, at the end of meiosis 1, we'll get two daughter cell with a reduced number of chromosome that will enter into the next meiosis 2 just after the cytokinesis. So in the beginning of the meiosis 2, two cells are there, and both of these two cells will undergo the process of the meiosis 2. And the meiosis 2 will be just like the mitosis process. That means it will include four steps, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. In prophase 2, in both the daughter cell of the this meiosis 1, the chromatine material will undergo the process of and it will attain the identity of chromosome. The nuclear material, nuclear membrane, nucleolus, then endoplasmic reticulum, Golgi apparatus, all will disappear. Spindle fiber will starts formation, and then it will enter into the metaphase 2, where all the chromosomes kinetochore will get attached with the spindle fiber and get aligned at the equatorial plate, and the which is attached with the spindle fiber coming from two opposite poles. Now it will enter into the anaphase 2, where the centromere will get splitted and two chromatids and half of the kinetochore or this centromere will be pulled apart towards the two opposite pole, north pole as and south pole, because the spindle fiber is getting reduced in size. Then after all the two sets of chromosome will be aligned, reached towards the two opposite poles, and one cell has two set of chromosome. Both the cells have two set of chromosome on two opposite poles. Now the cell will enter into the next stage, that is telophase 2. All the phenomena which occurred during the prophase 2 will now be becomes reversed. That means the chromosome which was undergone the process of condensation will undergo the process of decondensation. It will lose its identity and it will occupy the structure of shape of chromatine material. After that, the nuclear membrane will reappear, nucleolus will reappear, endoplasmic reticulum, then Golgi apparatus all will reappear, and both the cell having two set of nucleus. Again, after the formation of two set of nucleus inside both the cell, they will undergo the process of second cytokinesis, and here also at the end we'll get four cells from the, that means two cell from one cell and two cell from one cell. In this way, at the end of this meiosis, we'll get four cells having half number of chromosome, and this is the special feature of this meiosis, to maintain the chromosome number from one generation to other. In this way, meiosis is very significant, because during the meiosis only the chromosome number get reduced to maintain the chromosome number, as well as during the recombination process, because the chromatine material, the part of the chromosome, the genetic material is get exchanged. That's why the variation will occur. So variation will, speciation will, all these things will depend upon the meiosis, the process of recombination. In this way, meiosis is very significant. At the end, by this we came to end of this meiosis, and all these steps we'll discuss through the diagram also. This is the second type of cell division, that is meiosis. Now meiosis consists of the following stages. It has two parts, that is meiosis 1 and meiosis 2. Meiosis 1 has prophase 1, metaphase 1, anaphase 1, telophase 1, whereas meiosis 2 consists of prophase 2, metaphase 2, anaphase 2 and telophase 2. At the end of this telophase 2, there will be 4 daughter cells with half number of chromosome. That's why it is also known as reductional division. Now this prophase is the longest phase, having the 5 stages that we'll discuss. Then this is the diagram which is showing different stages. You can see in this diagram how these sister chromatids separate and the haploid daughter cell will form at the end. Now we will discuss about the prophase 1, all the stages, in the next slide. This is the second type of cell division, that is meiosis. Now meiosis consists of the following stages. It has two parts, that is meiosis 1 and meiosis 2. Meiosis 1 has prophase 1, metaphase 1, anaphase 1, telophase 1, whereas meiosis 2 consists of prophase 2, metaphase 2, anaphase 2 and telophase 2. At the end of this telophase 2, there will be 4 daughter cells with half number of chromosome. That's why it is also known as reductional division. Now this prophase is the longest phase, having the 5 stages that we'll discuss. Then this is the diagram which is showing different stages. You can see in this diagram how these sister chromatids separate and the haploid daughter cell will form at the end. Now we will discuss about the prophase 1, all the stages, in the next slide. This slide is showing the 5 different stages of the prophase 1 of meiosis 1. Now the first stage is known as leptotene. In leptotene, the nuclear membrane will undergo breaking and finally it will disappear. Before that, this condensation of chromosome will take place. That means the chromatin material will attain the shape of the chromosome and it becomes shortened in length as well as acquire the shape of chromosome. Now in this way the chromosomes will show its 2 arms known as bivalent, and these 2 non-homologous chromosome will undergo the pairing in this way, and this phenomena is known as synapsis. Here we can see this is known as the synaptonymal complex which occurs between 2 non-homologous chromosomes. Now so the synapsis is the identifying feature of this stage, that is zygotene. Then it will enter into the third stage, that is known as pachytene, where these 2 chromosomes will exchange their part at that particular point. This site is known as crossover site and this phenomena is known as crossing over, and this requires one enzyme known as recombinase. Then the next step will come, that is known as diplotene stage, where the chromosome which are forming the synaptonymal complex, these will separate, that is dissolution of synaptonymal complex, and they will get separated only at except that point of crossing over, and that is known as chiasmata. So this chiasmata is the identifying feature of this stage, that is diplotene, and now the cell division will enter into the next stage known as dikinesis. In dikinesis, the chromosomes, that means terminalization, this chiasmata will undergo the process of terminalization, that means it will move towards the terminal end, and finally these 2 chromosomes along with this exchanged part get separated, and this is known as the dikinesis.
So, terminalization is the identifying feature of this stage, and now finally this nuclear membrane, nucleolus, all these things will undergo the process of disappearing, and these chromosome will come inside, direct inside the cytoplasm, and then the prophase 1 completed. Now the spindle fiber will take place like mitosis, and then the cell will enter into the next stage, that is known as metaphase 1 stage. In metaphase 1, the again the same way, that is at the equatorial plate, the cell will, all the chromosome will get attached, arranged. Then anaphase, the division of the kinetochore along with the two arms of this chromatid will be get pulled apart toward the two opposite poles, and finally in telophase, two sets of chromosomes or two sets of nucleus will, two set of chromosome is there, and that will form two nucleus inside the cell, and that will be followed by the cytokinesis. Now these two cytokinesis, after cytokinesis, these two daughter cell will enter into the next meiosis 2, that is just like the mitosis will undergo 4 stages, that is metaphase, that is prophase 1, prophase 2, metaphase 2, anaphase 2 and telophase 2, and the same type of event will be there, and these two cells will give rise to two cells at the end, and finally will get 4 cells with haploid number of chromosome, and in this way the meiosis will complete. In this slide we can compare between the different stages, that is metaphase and anaphase of the equational division, or this is the also in case of meiosis 2. Now in metaphase, all the chromosomes are present at this plate, that is known as equatorial plate, it is also known as metaphase plate, whereas in anaphase the each centromere divides, and the new chromosome will, each derived from one member of one set of paired chromatid, begin to move towards the both poles, that is north pole and south pole, and in this way we can see this is the comparison between metaphase and anaphase. This figure is showing the metaphase in mitosis, this is metaphase 1 in meiosis, and this is metaphase 2 in meiosis. We can see in almost all these cells, this equatorial plate is there where the chromosomes are get arranged. This slide is showing the diagrammatic representation of the event cytokinesis. This is animal cell in which the cytokinesis takes place by the formation of this furrow, that is known as cleavage furrow or the groove, which formed at the periphery, and it will move towards the center, and finally it will give rise to two daughter cell. But in the plant cell, the cell plate formed in the center, and this will move towards the periphery and finally cut this cell into two, and at the end will get two cells, daughter cells from the parental cell, and this is showing different types of the method of phenomena of cytokinesis in animal as well as plants.